Hello, everyone, and welcome to Heads Up, the weekly webcast and podcast of the National Headache Foundation. I'm Dr. Lindsay Weitzel, founder of the Facebook group Migraine Nation and chronic daily migraine survivor. I am here today with Dr. Tim Smith. Hello, Dr. Smith. How are you?
Doing well. Thanks for having me on again.
Thanks for being here. Many of you know Dr. Smith. He is a regular on Heads Up. Dr. Smith is the CEO of Study Metrics Research, and he is also the Vice President of the National Headache Foundation. So today we have an awesome topic that is one of my favorite topics related to migraine. We're going to talk about genetics and migraine. We're going to discuss a paper that was recently published in Nature Genetics that discusses 123 genes or genetic loci that were found to be associated with migraine, which is really exciting for us because it opens the door to discover some pathophysiology related to migraine, drug targets. Anytime we find genes related to our disease, this is great information for us. So let's talk about this paper. I invite everyone to just think about all the people you're related to that have migraine, including you, because this is important for today's topic. I have my father has chronic migraine. I have chronic migraine. I have a son who was diagnosed with chronic migraine when he was 7. So many of us are fully aware that this is a genetic disorder. So get comfortable and let's ask Dr. Smith some questions. So let's be honest, there's a lot of stigma associated with our disease. And I honestly feel like these types of studies validate us a little bit because we don't really have genetic markers of migraine. So what do you feel, Dr. Smith, is the best use of these gene studies?
So the gene studies of the type we're talking about today are, they're really great for identifying some target markers, right. New genetic and hence molecular, because all of those gene aberrations that are associated with migraine attacks code for proteins that make up cellular functions and vascular, targets. Mm-hmm. And so it's basically a big hypothesis generator. So we have 123 new risk loci in our armamentarium, or what we know about genetic linkages to migraine. And so these are a lot of hypotheses that we can look into to develop new treatments, possibly for helping us select treatments, because as you know, most of migraine care now is trial and error, and this might help us be able to zero in on effective treatments. More likely, I think we still have ways to go to be able to do that, but that's one possibility that could come out of this.
Right. So migraine is described as a polygenic disorder. Can you talk to us about what that means?
Sure. So polygenic, the literal, Latin translation, poly is many, and genic means genes. So basically, it means that there are a lot of different potential genetic causes, different gene mutations that can lead to a problem in your neurovascular system that give us what we call migraine. So there's the phenotype, which is the symptoms that people have, and the genotype, which is the DNA abnormalities that code for the problem. So genotype versus phenotype, and we may have different phenotypes, but there are some central symptoms of migraine that we recognize across the spectrum of migraine. Mm-hmm. And so a polygenic disorder like migraine means there may be different DNA mutations that lead to the phenotype of the migraine attack.
Okay. So is this part of why migraine is so diverse? Why my migraine doesn't look like your migraine? And is it because we might have a different genetic makeup that causes our migraine disorder?
Yeah, I think that probably has a lot to do with it. It's not proven, but it sort of makes sense, that while we may have some central features that are alike across the population of patients, there may be some differences, like the difference between aura and not having aura, or patients who have they have more gastric symptoms than not, or you could make up whatever different kind of presentation you want. And what we don't know is if it has anything to do with propensity for increased frequency or susceptibility to medication overuse headache,
Right.
or chronification, or whether they go away, whether they're hormonally related, whether they get better with age. There's all these different things, and patients present with different stories, and some of that may be determined by some of these different gene mutations that may account for all the different presentations.
Right. So this study performed something called a genome-wide association study. Can you give us a bit of an explanation of the strengths and weaknesses of this type of genetic methodology?
Well, so what they do, it's a great big data mining exercise.
Mm-hmm.
Basically, they have a huge pool of genetic sequence, basically patients who've had their genome sequenced, and they used 5 different databases and combined all of these different gene sequences from over 100,000 migraine patients and 700 something thousand controls. Mm-hmm. And when you have that large of a database and something about the patient's history, that is whether they have migraine headaches or not, then you can match up what kind of headache they have, what their migraine attacks look like, and we can compare that to these genetic sequences. It's not exactly needle in the haystack, but it's looking for any genetic sequence that tends to be associated with migraine. And when they did this analysis, there were 123 of them that kind of rose to the top out of thousands and thousands. So it basically brings forward this hypothesis that these genes may be part of the polygenic makeup of migraine in a population of patients.
Okay. When they use this methodology, were they able to discover the function of each gene that was found? Is that something that we know?
No, this just only gives us the association. So
okay.
In research, we have a saying, association does not equal causation.
Right.
But that's why I refer to it as a hypothesis generator. But when we looked at this, it was interesting that one of that we've only proven 3 gene associations with a specific subtype of migraine called hemiplegic migraine. Mm-hmm. And we know what those genes code for and what's made abnormal by those genetic mutations that lead to the hemiplegic migraine. It's easily recognizable because of the way it occurs. But presumably there are genetic abnormalities that can lead to other protein aberrations, maybe an ion channel, an ion pore that doesn't function right, that allows the brain to be more sensitive, or our blood vessels to react in a certain way, or nerve, pain signals to be transmitted more efficiently. And one of the ones that was associated with migraine with aura was one of the known gene abnormalities for hemiplegic migraine, which is a type of migraine aura. It's not very common, but when you see it, you recognize it. So that's kind of validating there. But for all of the others, we don't have that known, we don't know what those genetic sequences do and then how it behaves physiologically. It can be studied, but what this can do is kind of give us a little more of a laser focus on some gene aberrations that could be studied, and we could open up a whole new world of diagnostics and possible therapeutics for those patients that experience a particular headache type.
Right. Using your previous terminology, we don't know exactly how this is manifesting phenotypically or clinically, just when we have these genetic aberrations. One of these things, the things I find exciting about this study and these 123 genes, 86 of them were previously known to us from a study done in 2016, but that means 38 of them are brand new to us, right? So first of all, what I'm curious about is, do you think we found them all? Or do you think we might find some more genes related to migraine in the future?
Right. I don't think we have found them all. I don't know if we've even scratched the surface. This could be the majority of them. What we know is, the difference in the two analyses that you mentioned was that the first one done a few years back was on a smaller set.
Right.
And then what these investigators have done is they've gotten a much larger data set from these different databases where people would have their genes sequenced. And these are companies like the 23andMe was one of the companies that they got a data dump from, and there were 4 others. And so people who volunteer and they give part of their history and they tell what their medical diagnoses are. And then when we go through and do these sophisticated data visualization studies, then we can see which of those they call them risk loci. So these were small bits of genetic material that had a unique pattern that was associated with migraine. And so no, we don't think this is all of them. I think if we continue to add more and more to the database, we're going to see more and more potential risk loci that are associated with the different migraine phenotypes.
Okay. So we learned a few really interesting things about subgroups. So one of the things the authors looked at was whether they could group the genetics of migraine based on people who had migraine with aura and people who had migraine without aura. What did they find there?
Right, so there were a small number of these risk loci, they call them, there were 3 that were highly specific for migraine with aura, and there were 2 that were highly specific for migraine without aura, and that meant that it was almost exclusively associated with the migraine without aura and not with migraine with aura, and vice versa. So that was sort of interesting, and it would suggest that people who have one of those risk loci would have migraine with aura, and then the converse would also be true for the other group. But interestingly, there were nine risk loci that were associated with both,
right,
equally. So it remains to be seen what that means, but I don't think it necessarily means that if you had one of these that you could reliably predict that this would be something inherited through your family.
Right.
We do know that that does happen. And for example, hemiplegic migraine, when it occurs, you can do a pedigree and you can see where the cases come through through the generations. So that could be the case on some of these, but I don't think we can hang our hat on that just yet.
Right. So I was going to ask a question just to make that, more real world for us. Does that mean if my mother, let's say, had migraine without aura, would it be impossible for me to inherit migraine with aura?
Right. No, it would not be impossible. If she had one of those overlap ones, then we see it go both ways. And then you only get half of your genetics from your mother and the other half from your father. And so you could have a different expression. And certainly we see some people in families that have migraine with aura and some that don't. Right. And there has been debate, though, I think, throughout, the last couple of decades, really maybe 3 decades, as to whether migraine with aura and migraine without aura are the same thing or if they represent different entities. And I think what this analysis shows is that there are probably some folks with migraine with aura that is a different entity than, when you look at the polygenic, potential for this, there some patients are that they do have that risk that lines them up with migraine with aura, and then others that do not. But then there are those overlap ones too. So it doesn't throw it completely out the window, but you can't hang your hat on it either.
Right. Then my next question is, there's always been some debate within the migraine community and the medical community about whether migraine is a neurological condition, whether it's a vascular condition, and then we all just call it often a neurovascular condition. And they tried to look at that based on these genetic loci. What did they find there?
They found that that last term you used was probably the most appropriate, the neurovascular. It's 50 years ago, physicians thought of migraine as strictly a vascular phenomenon. And in the 90s, researchers identified these the trigeminal nerve activation and the inflammatory plasma extravasation of the inflammation of the dura and the lining around the brain as mechanisms that didn't involve blood vessel changes.
Right.
And so there was this increasing school of thought that it was a pure brain disorder, just a neurologic, and that the vascular changes that we saw were reactive to the neurologic phenomena that were going on. And these risk loci coded for proteins that were highly specific for neurologic tissue and vascular tissue, and not much else in the body. So that really kind of it's a big broad strokes way to refer to it, but it looks like it's easy just to separate it out as neurologic versus vascular. It's both.
Right. And how do you think we can use this gene data to improve the lives of those of us with migraine? What do you think is how do you think we're going to use it?
Well, it gives us a whole lot of a great big number of new hypotheses, new potentially molecular targets to research. And I think it also explains why there's not a one size fits all treatment, for this. And so we can understand, and unfortunately for many people, they don't respond to a number of things. And it may be that they have a very unique genetic abnormality that makes their migraine behave differently and respond differently. Yeah. So I think we have hope of new molecular targets that might come from this, that might help those people that don't respond to the things we have today. And the other thing is, I think I mentioned it a little bit earlier, is sort of trying to predict responses to certain therapies instead of just being a trial and error kind of thing. So if we knew that someone, and one of the mutations that they found was actually highly specific for a CGRP gene, and we know the CGRP monoclonals and the GPANTs block the effects of CGRP. Right. And it may be that if we could develop, I could see somewhere down the line having a blood test that would tell us whether or not we should be a CGRP blocker responder. Yeah. So it could be interesting if it turns out that way.
Right. I love that idea. I love the idea of using this data to make new medicines. I love the idea of it helping, hopefully there being better treatments for those of us that have children with migraine in the future. I want it to be a better world for them. And I love the idea that it helps us know, possibly someday it could help us know whether or not we're going to respond to a medicine. Is there anything you'd like to add to our discussion on this awesome genetic and migraine data?
Well, I guess, the one big observation that people like me would take away from this is that this whole concept of looking at real world data and doing data mining from these huge databases and doing these matching exercises so that we can identify associations that could lead to new discoveries and new cures. It's pretty fascinating. And just a few years ago, we didn't have these capabilities, but now we have supercomputers and huge data platforms that can, we're talking about columns that are in the millions, as
Right.
opposed to, if you took a stats class or something when you were in college, you might have worked with these tables, and the more columns they had and more rows they had, the more complicated they were to solve. And now we have the supercomputing capabilities, the AI and other machine learning capacities that we can put to use for us and analyze huge, unbelievably huge volumes of data and come away with learnings that could be quite significant. And you combine that, and the other thing that's enabled this is the mapping of the human genome, which was accomplished a few years ago. And when the human genome was mapped, we all said, "Oh, now we're going to learn so much. Cures are just going to come falling through the rafters, and we'll have it's going to fix everybody." But it's been a little slow and cumbersome getting there. But I think what we've needed is these large databases to be put into systems where they can and use software packages that are capable of doing these huge analyses and giving us understandable results. And that's kind of where we're going and where we are these days. So I would suspect that these kinds of analyses may become more and more commonplace as we go forward, as we have more and more patients that have had their genome sequenced and contributed to databases where this kind of analysis can be done.
Right.
Pretty interesting.
Right. Well, thank you so much, Dr. Smith. This was a super interesting webcast and podcast. And thank you, everyone, for joining us this week on the weekly webcast and podcast of the National Headache Foundation.